Hi, and welcome to How Plant Scientists Work series at Plant Biology 2016 in Austin, Texas. I'm Ian, and I'm here with Annie from NASA JFK, and he's going to tell us a little bit about how he works. So, hi Annie, tell us about yourself and your background a little bit.
So, as he mentioned, I'm a plant molecular biologist. I'm part of the NASA postdoctoral program at the John F. Kennedy Space Center in Merritt Island, Florida. And I work with Arabidopsis, and my project explores what are the effects of elevated and super elevated levels of CO2 on plant development, and I'm doing more work on that regard.
Okay, so I think that covers the where you work and your specific title and role. So what's one word that describes how you work?
Let's say space life sciences.
Space life sciences, okay. Going for the big adjective. All right, and what's the favorite thing that you do at work?
The favorite thing of mine is growing plants. And the best part about that is because I get to use all the latest things and all the environmental chambers that can do a lot of crazy things. So I'm happy with my plants there.
Like what? Like what's one crazy thing?
Well, the one crazy thing is because I work with elevated and super elevated levels of CO2. So just to give you an idea, the current the CO2 levels in this environment is only 400 parts per million. That's what we breathe. And as you know, the global climate change, the CO2 levels are constantly increasing. But in my work, I deal with CO2 levels ranging from 1500 parts per million all the way up to 15,000 parts per million. And that too in small confined spaces. So if I'm not polluting my own experiment, I'm wearing a mask which takes the CO2 that I'm breathing out of the growth chamber. So I'm like constantly doing experiments in an enclosed environment with a tube on my mouth that takes the excess CO2 out so that I don't pollute the environment.
Okay. And do you have a favorite plant? Is it Arabidopsis?
Well, yes, because Arabidopsis is the best thing, I guess.
All right. That doesn't have to be like. I mean, it can be an aesthetic pleasing choice.
Well, I would say lettuce is pretty decent because they could eat it up there. So I'm saying Arabidopsis #1 because
up there being the space station, the International
Space Station. So yes, Arabidopsis #1, lettuce #2, and then plants, of course.
Okay. And I think you already mentioned one specific project you're working on, the highly elevated CO2 and plants' response. But do you want to say a little bit more about it at all?
Sure. The reason that we are looking into it because the idea is to grow plants for space environment. So as Ian mentioned, so International Space Station is the first thing, and this is not a science fiction. There are currently plants growing at ISS, and those are the lettuce plants that the astronaut recently ate as a salad. So the idea is to keep that going and have fresh food available for astronauts if you are targeting for the Mars mission that's coming up in 2018. So the idea is to have plants constantly growing for this environment, and they don't have to depend upon carrying so much of the dry food or food items with them for such a long trip.
Okay, and so let's get into the specifics of how you work. So what is your workspace setup like?
Well, I work mostly in the cell molecular biology lab as well as the environmental growth chambers lab. So environmental growth chambers, as I mentioned, I grow Arabidopsis plants that are exposed to various levels of CO2. Then I take those plants, go into the molecular biology, do a lot of PCRs, transcriptome sequencing with them. We have a proteomic sequencing also coming up next week. And then we look at the genes that are upregulated, downregulated, and the whole plant physiology level changes. And also in the growth chamber lab, we look at all the physiological parameters like stomatal conductance and optimize all the growth parameters there.
Okay, and how do you learn new things?
So as I mentioned, my training is mostly in plant molecular biologist, but over there I'm working with people in chemical engineering, industrial engineering, hardware development. So every time I'm working with somebody, I learn new things, like this development or the construction of new growth chambers, which I had no idea about. So I just went ahead with them, learned how to construct these growth chambers, how they control different parameters, what are the sensors for those growth chambers. So I'm in a different zone over there when I'm learning all these new techniques from these people.
Okay. And the last one is, how do you work on projects with colleagues or teams or groups? Well, sounds like you do that a lot at NASA.
So what happens is usually we have weekly meetings with all these people from different groups. We come together, we update each other, we give us updates about what difficulties we are facing. Maybe I don't know things that they are doing, and so on and so forth. So we exchange ideas. Like when we were building growth chambers, we didn't know how to control the CO2 levels because there were mechanical scrubbing and chemical scrubbing of CO2 excess. But then we figured out a way that we could have sensors, and those sensors should be controlled by computers, which can add or remove CO2 at the same time just to keep the levels balanced. So, but just by interacting, and of course my boss is a horticulturist, so he gives me a lot of input about the whole plant physiological data. So that helps me a lot. So working with all this expertise gives me, or actually I learned a lot of things from these people apart from my own knowledge.
Okay. And then the last thing is, if you could tag someone to do this series next, either by video or the text version on the blog, who would you like to nominate to do these series of questions? You don't have to name anybody if you don't have anyone in mind.
Yeah, I'm
okay.
completely blank about it.
Okay, that's fine. And thank you.
Oh, thank you very much. Appreciate it.
Yep.
Thank you.
All right.